So for those of you that hadn't been able to join us in the session earlier with Dr. Jack Miller, once again, good morning and good afternoon or good day, depending on where you are across the globe. Welcome to today's live events at ASBS 2020. We had a lot of really interesting talks already, and if you haven't been able to, go to our YouTube channel. I will put the link into the chat in a moment. And today's session, just as all the other live sessions, will be recorded and uploaded shortly after we finish. And if you have any questions during the talk today, please post them in the Q&A, not in the chat, but in the Q&A, so we can answer them live here with our panelists. And with that, it is my great pleasure to introduce our two speakers this afternoon. Or at least it's my afternoon, I apologize, I don't think it's anybody else's. We have Dr. Sylvain, who is the principal investigator in the Space Biosciences Research branch of the NASA Ames Research Center and Genelab project manager. He received his two masters actually in physics, mathematics, and nuclear engineering in Grenoble, France, and at Texas A&M. He also obtained a PhD in nuclear engineering from the University of California, Berkeley, and then did a postdoctoral fellowship in quantitative radiation biology at the University of California, Berkeley. He is now the project manager of Genelab, coordinating the systems biology scientific research effort for Genelab, for any of the Genelab projects. He's supporting internal and external collaboration efforts and is the lead of the Radiation Biophysics Laboratory. Upon joining the Lawrence Berkeley National Laboratories in 2004, he led computer modeling efforts for NASA's Specialized Center for Research, looking at risk for space radiation in human breast, and supervised the bioinformatics lab for multidimensional microscope image analysis, agent-based modeling, and genomic analysis. So we're really looking forward to your talk, telling us more about Genelab and the whole concept that we can find. Our second speaker following right afterwards is Samrawit Gebre. She is the Genelab Deputy Project Manager at NASA Ames Research Center and is a biotechnology professional with experience in bioinformatics, data curation and analysis, and project management. She currently provides technical management support for the Genelab project and leads the data curation program for data import, submission, and management of the Genelab data repository. So please join me in a warm welcome to our speakers that share their insights into NASA Genelab, the NASA Systems Biology Platform for Space Omics, repository analysis, and visualization. Thank you, and please take it away. Sorry, Sylvain, you are muted, I'm sorry.
Still working now?
Perfect, yes.
Okay, so thank you for the introduction. Let me jump straight to the platform Genelab, the NASA Genelab, which is the NASA System Biology Platform for Space Omics Repository Analysis and Visualization. So you all have seen this graph. It's the classic NIH cost of DNA sequencing per megabase as a function of time. And for a long time, I mean, for at least, from 2001 when the human genome was first cracked to 2007, the cost was pretty much following the cost of computer, industry, called the Moore's Law. But in 2007, 2008, as you know, the way sequencing was done was changed and really made us go up this curve. And then since then, the cost of sequencing has been going extremely low. And so the human genome today is affordable. It's about $1,000, even less now. In 2011, there's something called the Decadal Survey. So that's the NASA has this, the National Academy of Science gets together to decide on the next 10 years priorities for the for the agency. And so the decadal survey back in 2011 said that all the omics techniques were going to be essential to really better understand what's happening in space. And that was done when the cost of sequencing was still fairly significant. Move forward now, 10 years later, well, first of all, that decadal survey really triggers the interest and the creation of Genelab in 2014. And we're now in 2020, and the cost has dropped probably another 100, at least 10 to 50 times. So what was relevant 10 years ago is even more relevant today. This is a classic slide that is from, more focused on the human research project. And I have to say that Genelab is not funded by HRP, the human research program of NASA, but we are funded by space biology. So they have different emphasis. We don't emphasize as much the human side. Having said that, everything we do is supposed to inform in some fashion what's happening to the human. So let me give you the bigger picture here. Those are the 5 stressors that are known from space travel. Radiations, cosmic rays, gravity field, so loss of gravity is a big one. Closed environment, confinement, and distance. This is really relevant when we go to the moon and especially to Mars, where communications are very difficult. In this case, the type of endpoint you're going to see is limited healthcare, mental stress. So I'm showing you different outcomes from these different stressors. And some of these outcomes can be the result of multiple stressors. For instance, visual impairment is the result of radiation and gravity, loss of gravity. Other endpoints are very much driven by one stressor, like cancer has been primarily driven by radiation. Bone loss is a combination of radiation and gravity field. So it gives you an idea of the complexity of what we're looking at. There's multiple stressors and multiple endpoints. Now, if you step out of this picture and then let's look at the type of data we have, well, and how we can better archive and share and mine this data. There's really 2 legs. The yellow side here is the experimental model, and the blue side is the astronaut physiology and environment. So measurements we can do on the astronauts and in the environment of the astronauts. And so the goal of the scientists is to really try to put this data together. One experiment alone doesn't tell you much. A lot of experiments together should give us the big picture and really help us. So, in terms of experimental model, you can go on the ISS and simulate and really have true, space environment. Or you can simulate radiation on the ground going to Brookhaven National Lab at the NASA Space Research Lab, where basically you can simulate cosmic rays on samples. There are very different type of exposure. In space, you get a chronic exposure, and on the ground, you're going to get an acute dose. So there are differences in these different experimental models. And then we can use different biological measurements and models like human cells, and big mammals like rats and mice, and you can go all the way to Drosophila and small animals. And you can play with the genetic diversity of these animals. So I'll walk you through this in a minute. And all these measurements can be used to model and predict risk of the life in space. On the human side, you really have this environment. So the environment is the radiation you have. Of course, we have a good idea what's the cosmic radiation on the ISS and on the moon. It's pretty constant. But then you have the solar particle event that can be something that changes very quickly. So, all these monitoring is important. You also have to monitor the inside of the capsule. And so this is CO2 level, oxygen level, temperature, humidity, light cycles. And then you have the activity of the astronauts, what they do for countermeasures and their vitals measurements, their pulse, their blood pressure, and so on and so forth. So all this together, the idea is eventually to really go after countermeasure and personalized space medicine that really uses all this information to infer on the fly what needs to be done to an astronaut to make them safer. So let's focus a bit on this animal model. So Genelab can take any kind of data, and a big chunk of it is animal. And on the animal, we really focused on the large animals. Typically are mice. We have a few rats data, but anything on the space station, for instance, typically it's mice for the biggest size. And then you have smaller animals like C. elegans and Drosophila. The smaller the animal, the more animal you can fly in one payload on the ISS. So that's the power of the small animals. And so you can really, tease in things like genetic diversity, knocking out a specific genes and looking at the impact of that gene, or having a bunch of different genes being knocked out, or having a different strands and do a GWAS, for instance. Okay, on the mice it's much harder because we only can fly 20 to 40 mice in one payload. Of course, the mice and the bigger animals are more relevant to the human question because their response and the ortholog between the genes of a mouse and the humans are pretty close. And so eventually we do want to extrapolate to the human. The fly system, actually, the Drosophila, is a great model. It's been shown to be a good model for circadian rhythm, oxidative stress, neural behavior. And so a lot of GWAS studies have been done already to model some of the human response using Drosophila. So a fly, even though the genome is 18 times smaller, can still tell us a lot about humans in space. The other type of data we collect is microbes. And the microbes here can be microbes being flown and culture in space, or it could be microbes that we discover in the capsule using swabbing techniques on the walls and seeing what grows and what prevails in this environment. Of course, we also have plant experiments. You've all seen these great movies of the growth of plants and how the lack of gravity affects the root growth and the light and so on. So we have this kind of data as well. And then a big emphasis in Genelab is to really try to think of this as an ecosystem, all these things together, working together, interacting with each other. And really, for this aspect of linking the data, we really need multi-omics and system biology. So let's dive into Genelab. What does it look like? It's a cloud structure. We on Amazon Cloud. The data are stored there, and then if you were to come on our website, you would see 4 different portal, if you want. A submission portal, which I'm going to walk you through in a minute. A data workspace, which is highly linked to the submission portal, is where you put your data, where you can share data with others. So it's kind of like a Dropbox, if you want, but for omics, very much focused on omics. And we have no specific omics that we take every omics. And actually, we even take non-omics data at this point. We have, of course, the data portal. This is probably where the majority of people go to. This is where the data are. And then we have the analysis tool shell, which is a tool used to teach bioinformatics and working through our data. Let me walk you through the submission portal. So as a PI, you will, any space bio PI needs to submit their data to us. Any omics data should go to Genelab. And they can submit it through the submission portal that Sam will demo in a minute. So I'll let her walk you through it. We also have, this is something really unique to us that you don't see at NIH for other databases like GEO or SRA or TCGA or whatever. We produce our own data. And then doing this, we have the Genelab now has a fairly large team of technicians that are actually very well trained to process samples from the space station or any samples really. And process them in a way that is very consistent. RNA is just not RNA. There are different prep that can really affect your sequencing, for instance. So we are, just like NIH has done with the Cancer Genome Atlas, we are trying to really standardize the approach as much as possible. And we have put in place a system that really streamline this and make it very robust. And so whenever an experiment is done in space, a PI may have written a grant on the mammary gland. Well, there's a bunch of other tissue than the mammary gland in the mice. So when they come back, they take their tissue and then the rest is going into a sharing process called the biospecimen sharing program. And Genelab is one of the big users of the BSP because we request competitively some of the tissue and sometimes we get granted the tissue and we can run and augment this experiment by adding more proteomics and transcriptomics and epigenomics on the samples, for instance. One thing that happened is because we have set up standards that actually we now are imposing in terms of omics for our PI, a lot of the PI actually end up using us now to process their sample. It turns out to be better done and more consistent. And we encourage all PI who wants to have data that they can compare to a large array of space omics data to use us. Right now, it's highly focused on, on NASA PI. Mechanism for outside NASA PI needs to be worked out. Okay, let's now go to the data portal. The data portal really has three types of data. The raw data, so those are going to be your classic FASTQ5 or RNA-seq, really the raw data. But we also have process data, and of course, metadata are critical. They describe the context of this experiment, the environment of the experiment. So the process data is fairly new, and you can actually, so the metadata, for instance, all the environmental data can be visualized now with a visual tool that you will be demoing in a minute. So that's, for instance, the radiation on the space station. You can actually visualize the response as a function of time during a mission. You also have a visualization portal that really lets you see the process data. And so those process data are, from raw data to what are the pathway modified in space for a run-on research mission. That's the kind of visualization you can do with us. You'll have a demo on that soon. On the analysis tool shed, this is really an online tool called Galaxy. We didn't create that, but we adopted it and we improved it in a way and really made it focus to space omics. And it's really interesting because now we have written tutorials, we have script that you can use and pipelines that are really based on the standard we put together. And anyone can go, and universities are starting to use us to really teach bioinformatics and make people discover space omics, which is not something people know, and they get really excited about it. And together, this is really kind of like a citizen science project in a way, where we have all these people mining the data and gaining new knowledge. So I call that data democratization. We initially only were storing and archiving raw data, which were really difficult to interpret. But by processing the data, we really made this data available to anyone. And so this new knowledge really is available to now any citizen scientist. It's not just limited to the data scientist or the biologist to a certain extent. In this process, I want to focus on two groups that have been essential for this work: the analysis working groups and the education working group. So let's start with the analysis working groups. We have about 120 people who have joined us working on a volunteer basis with us. We have an annual workshop every year to basically go over the data and come up with solutions. And we meet monthly in 4 different groups focused on the groups I mentioned earlier: animal, multi-omics, plants, and microbes. Many different countries are join us. So please, since I know I'm talking to a large Australian community right now, we are welcoming you. Contact us and we'll be happy to put you in one of these groups and participate. So what's in it for you, since it's voluntary basis? Well, a lot of things are in it for you. We publish a lot of manuscripts together by taking the data and taking multiple data sets and creating new stories. By putting things together, we see new ideas and new concepts that were not detectable with only one data set. We also have come up with a way we have now pipelines that are a scientific consensus that tells you exactly how to derive the raw data to process data. So, so we're writing manuscript about this processing pipeline. And then you also get acquainted to all the research that's done. And so it's an opportunity for you to write proposal that can get funded by your local space agencies. So a lot of benefits in joining us. So this is the link for genelab.nasa.gov is our website for Genelab, and then if you go to AWG and join, you can join us. The EWG, which is the Educational Working Group. So by the way, so all the processing that we have done in Genelab was done through the pipeline that we agreed upon with all the members of the analysis working groups. It's very important. We did not come up along with this. We have consensus. And actually, there is a paper. There are papers now out that that you should look up from our work. It's on our website. The EWG basically is benefiting from those scripts, those pipelines, and also our analytical toolshed, Galaxy, to teach bioinformatics. The idea is that by having young individuals, high school, so we focus on high schoolers, undergrads, and graduates, so it's different tier level. And at each level, they can do a bit more difficult activities, but all of them get really acquainted with space biology. And it's a great way for us to we're kind of like the ambassador of space biology at this point for the scientific community and the world in general. So it's very exciting to see that the AWG is really gaining a lot of momentum. Universities are starting to use our data and our tutorial to teach. San Jose State University is one of the universities now that are using our system. We have interns coming every year. This year, because of COVID, unfortunately, it's not going to happen, but usually we have interns. Contact us for more information. This is the growth of Genelab over the past 5 years. You can see how we really have generated a lot of data. We have data sets and studies. The reason we have more data sets and studies than a study can have multiple tissue, as I mentioned. And so that's the difference between a study and a data set. We cover 45 different species. We have a fairly large database, 130 terabytes. Surprisingly, 54% is space flight, so ISS, satellite, that kind of data. But a big chunk, 44%, is ground studies. So those are typically analog experiments. So bed rest study for human. We don't really have astronaut data. We can't. At the moment, it's somewhere else. But we do have human data. Typically, bed rest study would be one of them. And for animal work, it's typically cosmic radiation and simulated microgravity. As I mentioned, we cover every organism: rodent, microbes, human, plants, invertebrate, fruit flies, fish, you name it. Assay type, all omics are welcome, no limit. Having said that, I would say because transcriptomics is such a mature field, 70% of our data are transcriptomic, 43% are microarray and 27% are RNA-seq. The RNA-seq is the growing part of the pie. There's not very much microarray done anymore because there's some issues with microarray. It's really hard to compare multiple data sets. So for that reason, I think most people have moved to RNA-seq. Something interesting that's a metrics for our stakeholders, but I think it really highlights some of the strategies and some of the key point that reflects the power of database. So we came up with that concept called return on investment. It's really a simple idea. So first of all, we have 300 plus studies, right? So you would think that each study has a manuscript linked to it. Typically, a PI submit a paper and submit their data into a website. We have to remember that Genelab was born in 2015 and really, we started ingesting data late 2015. There were a bunch of space bio microarray data that were there way before, and they were in Geo and other websites. So we ingested this data, but we don't consider this data, any manuscript linked to this legacy data are not considered original publication. We only consider original publication a PI who goes into our website and turn their data in and will submit their manuscript with the data in Genelab, which is the way they should do it because our metadata are much better than what you would see in Geo. We're just not archiving. We are doing more than archiving. We are curating very carefully, and any issue, any uncertainties is immediately reported to the PI, and we work with them to make sure the data are fully usable. And we also have a big emphasis on machine learning and AI-ready database. So we are considering that we have 47 original publications linked to our Genelab dataset at this point, and that 25 is a number of papers that were born from using the data in Genelab. So I call that the return on investment because basically, if you take the ratio of 25 over 47, it gives you, a fairly good outcome here, more than 50% return on investment. That means that with this database, we have multiplied our knowledge by 1.5. And I would even go further. I would say that those derived publications are even more powerful because they solidify something that was a hinge or an idea from one data set, which is statistically always weaker. But when you look at the group of data together, it's even more powerful. And we're growing very fast. So and a lot of these manuscripts actually are derived by the analysis working group. So another incentive to join us. Let me give you an example. There's a big chunk of papers coming out from the AWG this month in Cell. So in Cell Press. So there's a few papers that are going to be in Cell. Some will be in Cell Reports. Some will be in iScience. Some will be in Patterns. So Cell Press has a large array of different journals, Cell being the top one. So that one is in Cell. It's the multi-omics AWG paper. It comprises about 22 different data sets from proteomics, metabolomic, DNA methylation, RNA-seq. We use both human tissues and animal tissues. And actually on the human side, we were lucky to actually have true astronaut data. So those were, obtained not through Genelab, but they were really useful because they could confirm prediction that came out of the animal data and showing that the animal work is very relevant for the human questions. I really highly encourage that you go get this paper. It's in press, and actually you can see a sneak preview online. To make a long story short, the mitochondria seems to be disrupted in space, pretty much across all the different tissues. And of course, the mitochondria, the disruption of mitochondria in the liver, for instance, is quite significant. It creates a systemic web that really has implication everywhere. But it's not only in the liver. We also have effect at different tissue that we have looked at. So, and this disruption of the mitochondria, like I said, can explain many different phenotypes and different data that we have on humans. So, I really can't. This is a very long paper with many figures. It would take me an hour to just walk you through this story. So, it's a highlight. Those are the people that were involved. Again, you can see that it's involving everyone. People from the UK and I think Spain and so on. Okay, this other one is the plant analysis working group, and this is pretty cool. They actually took the challenge to take both macroarray and RNA-seq for transcriptomic on plants, and they classify them here with different colors based on the different type of hardware that we use: brick, petri dish, and so on, and also space versus ground. And of course, if you plot all this data together with the principal component analysis, if you throw all this together first, the obvious things happen. 83% of the principal component one separates the macroarray from the RNA-seq. No big surprise here. If we remove the macroarray and focus on the RNA-seq, then that's really interesting. You can start seeing really some systems, some plant, and some hardware either can show or sometimes do not show the impact of space. So again, I encourage you to go look at this manuscript. Okay, I emphasize the concept of standards a lot. And so one thing we're very proud to say is that, if you have heard about this, all the new databases need to be fair. FAIR stands for findable, accessible, interoperable, and reusable. And if you were to score Genelab versus the classic website like GEO, ENA for Europe, MGRAS, Metabolite, we have the highest FAIR score. Okay, full disclosure here, that score was, we evaluated all these databases ourselves, so someone may think that we are very biased, but I don't think so. I encourage that you go read this manuscript for, to think about it and let us know if you think that we were not fair to the other one, but I think it was a pretty accurate measurement. And it's not a big surprise because Genelab was created way after this other database. So we had the advantage of knowing what's expected. So for instance, we provide DOI for each data set, something that those websites don't do. So once you put the data in Genelab, you have a DOI, that means that your data are never lost. It will be, that digital object identifier will be there forever. And that's one of the critical functions for, or requirements for the findable part of a FAIR. We're using EasaTab, which is a very sophisticated metadata structure that is able to take multiple layers. We have a very advanced API. We're using MongoDB and JSON LD schema for classifying the metadata into objects, so you can do very advanced query, and really you can pull a query across different data sets, show me the impact of radiation on a tissue. So this is the power of FAIR, and we're striving to make it even better every year. And standard is important. As you can see, or as you can imagine, getting data in Genelab has a long road here, and each of these turns adds a layer of noise, from the preparation from spaceflight to being on orbit operation. Who's been dissecting the animals? Was the animal dissected in space? Was the animal frozen in space and dissected on the Earth? All these things, this tissue dissection, RNA extraction, the library preparation, all these features add noise and can literally change your data. And if you were to do principal component analysis, I can guarantee you many of these features, if they were done by someone else or if they were done differently, will separate naturally on your PCA plot. That's the reason why we are advocating for all the agencies to be very careful. We have so limited amount of data because it's so expensive to fly things that we should really have a strong consensus on the processing from A to Z. So to that extent, we have the analysis working group recognize this as being probably the most critical aspect of the data, and they created what we call the International Standard for Space Omics Processing. Please, this is the website. If you're interested, join them. It's an international group. We are not driving it. It's really driven by the scientific community. And you can have your say on how to process the standard, because I think this is going to gain a lot of, there are PI from JAXA, ESA, NASA in this group. And they have a paper out actually describing what they're trying to achieve. So it's not so much the standard, well, it's not so much sequencing or bioinformatics, but it's even before that, all the preparation of the tissue, how you prepare it. We discuss those things in these groups. It's a large group. It's very exciting. I really encourage you to join. Example of what the analysis working group has done is something more bioinformatic driven. This is our pipeline for RNA sequencing. And this is published recently. So it's actually in the Cell bundle paper that we just described. So I really encourage you, go look into Cell Press. There's going to be a lot of press release about those 20 plus paper that's going to be published. So plenty of reading to do. At Genelab, like I mentioned, we have our own sequencing group, and we really are now gaining a lot of requests from PI to do all the processing for them. Especially if you're a NASA PI, it's a great way to go about. And then we have the latest machine. It's a NovaSeq 6000. As you know, it's a high throughput. It's the best machine for Illumina, so we're very proud to have one of them. And on top of that, we have installed a lot of important standards, like we are putting ERCC spiking, universal reference RNA, and QC analysis at each step. So the universal reference is like typical tissue. I mean, either human cells or mouse cells are put in each run, so we can always standardize each run. I invite you again to go and read up on us about this sequencing group. This is the data that the sequencing group at Genelab has provided. So you can see we're providing a lot of data in volumes. Again, rodent research are the majority of our samples. This is actually rodent research mission 1. That's actually in collaboration with the ISS, and we're processing their sample as well. So not only Space Bio, but ISS is now working with us, and those are all the tissue coming up. We are pushing new technology like spatial transcriptomic and single-cell RNA sequencing. And actually, that's an example of a collaboration with a PI, Dr. Stefania Giacomello from Sweden, who developed spatial transcriptomic. She's one of the inventors of this technology. And we're very happy to work with her to really do both brain and heart tissue from animals that were flown in the space station. And you can see here how we can separate cell type and really have different features of the tissue as a function of space in this data. So more work is coming this way. There's no publication yet, but we're working on it. I think that's my last slide before I pass it to Sam. Just to illustrate the power of basically the analysis working group. We opened those group in 2018. You can see how our website has really increased the volume of session and users since then. And it's pretty much instantaneously the day we created this group. So you can see that they're very active and they're spreading the word. And the other big milestone was last year in 2019, we released our first visualization portal to visualize the process data that we had generated with the AWG. And then when we did that, we literally doubled the number of downloads. And that makes sense because we democratized the access of the data. Before, most of these folks were probably data scientists. Now, most of these folks are non-data scientists. They are probably high schoolers and undergrads and people who just want to mine the data. So that is the power of processing the data. On this, I will stop sharing. Let's see if I can do that. Share. Are you guys still there?
Yes,
yeah.
Thank you so much.
Okay.
That was fantastic. Yeah, I think we'll just hand it over to Sam if you want to share your screen. And thank you, Sylvain. That was really an excellent talk and an excellent overview. And I do want to say it's probably I think everybody else in the science is very jealous of this kind of standardization and having everything at once. Very brilliant and really beneficial for the system.
Thank you.
All right, take it away, Sam. Thank you.
Thanks, Chris. Thanks, Sylvain. Let me pull up my slides again. Okay, hopefully you can hear me and see the full screen for a quick little demo. I'm going to go over some of the apps that Sylvain described and show you a little sneak peek on what we're currently working on. So on our website, I'll go over how to search for data and what a typical study looks like in our data repository. I'll show you some of our features for our new submission portal. So if you have space relevant omics data, please stop by and happy to kind of walk you through how to submit data to Genelab. And then I'll give you a sneak peek on some of our new visualization portals that we're working on. So to get started, our website is genelab.nasa.gov, and this is what our homepage looks like. So on the very top, you can use the top menu to navigate through the different pages and different tools. So if you're interested in joining our analysis working groups, as Sylvain mentioned, please stop by, and this is a quick, easy way to join us. And then if you want to look at tutorials on how to navigate our website or how to perform RNA-seq analysis using our analysis platform, you can find tutorials under the help menu. In addition, if you are interested in Genelab, learning more, if you have any questions, you can also use the contact us under help, and feel free to write to us, and Sylvain and myself are happy to respond to any questions. So we do have our data repository, which holds all our data, but we also have all these supporting applications. So as Sylvain mentioned, we have our collaborative workspace, which allows you to store and share your files, and those can be associated to the submission portal to while you're submitting data once you are ready. And our analysis platform is a toolshed of bioinformatic tools that help you analyze your data, either the data you want to upload yourself or data that's in Genelab, and our repositories linked through that. So if you're interested in learning, please reach out to us and please look at that tutorial. And then I'll give you a quick overview of the visualizations that are linked both to environmental and our process data as well. So to get started, we're going to click over to our data repository, and this is a landing page that you get once you either use the top menu or the button I just clicked there. And here you can see that we have a listing of all the public data sets that are in Genelab. And on the right hand side, you can see that we have 284 public studies that are available for you to explore and navigate through. You can also use the search menu up top to search through the different data sets. So you can use the keyword search, you can use the search filters to search across Genelab. And if you're interested in the data sets that have process data associated and visualizations, you can kind of filter just to look at those studies as well. And for this example, I'm just going to use a keyword search. So in the keyword bar, in the search bar, if I enter mouse and liver and click the magnifying glass, you could see that we are reduced or filtered down to about 20 studies that have both those terms in the title, description, or part of organism and tissue type. A very unique feature to Genelab that is not across other kind of life sciences databases is the ability to search outside of Genelab. So what we've done here is we've federated 3 different databases. So NIHGO, EBI Pride, and MGRAS. So you can search across Genelab, or you can check the boxes right under the search bar and search across those other databases as well. So you could see here in my search that I have 2 that are from Genelab, as you can tell by our organism icons, or the other ones from Geo, for example, here, that have that icon as well. And if you click on this data set from Geo, it will take you there. So back to just searching in Genelab. As I go through the list, I find a data set or study that I'm interested in, and I just click on the title, and it will bring me to the full description of each study. As Sylvain mentioned, a big part of Genelab is really curation. We really focus on the metadata and making sure that we have all the data to continue analysis and store the information correctly for others to analyze, and even for our analysis working groups or our visualizations portal to really do a multi-analysis across data sets. So on our repository, this is an example of what a study will look like. As you can see on the left menu, you have different tabs or different sections. So you can navigate through that or scroll down the page to see the different sections as well. So just overall, just a quick description of each section. In the description tab, you'll find just general experimental information, including your description and contacts, high-level information about the experimental factors and organism and assays used. You will also find information that are relevant to the mission if it's a spaceflight experiment. And as you go through, you can record detailed protocols for each step. And we have a samples and assay table, and I'll show you what that looks like in just a bit, that describes the sample, that describes information per sample, and then has all the characteristics and parameters that should be recorded. If you do have a publication associated, you can have multiple publications per study, and those are associated here. Under our study files, you can download all the files that are, that are related. You can download one file at a time or multiple files, and I'll show you what that looks like. And lastly, if this data set does have processed data, it will have a visualization button, and that will take you to our visualization portal where you can kind of explore the visualizations that are currently available. Just some new features and some things that might be interested. If you are using the study in a paper or presentation, you can actually take the citation fairly quickly. You can download that citation or copy the information here under this box, and you can see that a DOI is associated with each data set. So you can properly cite a data set just like you would do a paper. In addition, on our samples table, we have a lot of metadata that goes on each sample, as you can see of how many fields this study has. But a feature on our website is the ability to choose the columns you want, and those selected columns you can export into a CSV. So you can filter down your columns and take that with you to do some further analysis. And that function is also available in our assay tables. Our study files. As Sylvain mentioned, each study could have multiple omics. So here you could see that this study has RNA-seq data, has proteomics data, and it also has some epigenomics data. So you can navigate through the different folders to see the files you want to download, and you can do a multi-file download. The example here is this data set does have processed data for the transcriptomics data associated. And what we do at Genelab is provide each intermediate step, so you can actually download the step that you're interested in. So you can look at normalized counts, so you can get right down to the differential expression table. And like I mentioned, you can select one file by just clicking on the file link, or you can click on multiple files by using the checkboxes on the side, and then using the download selected files button. This allows you to just download multiple files. Fair warning, some of the raw files are fairly big, so you might be interested in getting down to a little bit more of the process data to get you started. So that is a really, really quick overview of how to search for data and what a study looks like on Genelab. So, come through and check us out. On the other side, if you are a PI or have space relevant omics data to submit. I'm going to quickly go over our new submission portal, which is a really easy to use portal that walks you through the submission process to get data into Genelab and then publicly released when you're ready. So when you click on the submit data button, it will help you get a profile set up. And once you have a profile, the first thing you want to do here is create a new study. And when you create a new study, a really cool thing it does right away is provide you a GLDS number and a link and a DOI. So as you're populating your metadata and associating your files, you can actually just take this link and put it into your paper manuscript as you're submitting it. So you don't have to kind of wait for this link until you're done making it public. So that information is there for you right away, and it provides you guidance on how to cite it as well in your paper. So once you create your new study, we do have an option to go through a guided submission. So it provides you step-by-step workflow. And as you can see here, we have some hints that help you kind of know what to populate in each field. So those are available as you walk through. So you create your study, you're going to want to upload your files, and then we're going to jump into adding your metadata to your study description, and then more details on your sample and assay level. So first thing, we need title, description. You can add your authors here. You can add publications associated, and it's pretty easy. You could just use an add button here that's associated, or if you double click on the field, it's also editable. For some of the fields, we do have a way to automate auto-populate. So for example here, if I wanted to add a new publication and I had already a DOI associated with it, if I entered the DOI, you could see that it automatically populates all the other fields, so you don't have to do that. So those features are added in the submission portal just to make it a little bit easier for you to get your data in there. So once you have the information to get started, you want to enter your experimental factors and project details. So it's, once again, pretty easy to add. You can add new fields by using the add button here on the right, or you can edit the fields here by using the pencil edit mark here. So you want to add your factors, anything that describes your study design, and then some project details, and some of this is really relevant to spaceflight, but also includes like your funding information and acknowledgements and things like that. A unique thing to our submission portal is it supports ontology search, and we really do this to really harmonize our metadata across studies. And some fields do require ontologies. So for example, our factors, once you enter your experimental factor, and then you need to associate an ontology to it. So as you enter your terminology, it will start giving you some options for what term it should be. And for example, here, as I type in ionizing radiation, I could see that there is an ontology linked and a description that best fits that terminology. So you'll find that the first are usually the most common, and you can drill down through that list to find the right ontology that fits this term that you're looking for. So ontologies are required in most of our metadata fields just to ensure that harmonization. So we have our description and our overall experiment described. And as we get more into the details, we use the top tab here to navigate through and enter the information that's more specific to the experiment itself. So we like to have detailed protocols, so you can add new protocols. Depending on what assay you choose, there's always kind of a default template of fields that are already there, but you can always add to that by using the buttons here at the right, or you can edit the description as you get more. Another part of protocols and using this is a tab model, as Sylvain described, is the ability to add parameters associated with a protocol. So for example, for my sample collection, I wanted to add temperature. So when I edit the description, I have an ability to add a parameter here on the bottom left-hand side, and you can see that that's also associated to an ontology. So as I type, it gives me options on what ontology to choose, and you can select that, and what that will do is link the ontology to that term, and also provide that as you move over to your samples table. And you can select parameters for your assays tables as well. So it's how you add, important fields on how the experiment was conducted in a table format. So that is an option there as you build your protocols. And then that's linked to your samples and assays tables as well. As we navigate through the top tab, the next option is samples. And like I mentioned, each, depending on what assay you selected, there's a default set of parameters and columns set here, but you can always add more. So you can use the top menu of buttons here to add experimental factors for the ones you listed before, add characteristics that support and describe your sample itself. You can add samples, which will add rows to your table, and add additional parameters and comments. So you can, add more than what the default table has. And as you start editing the fields, you'll see that some of the fields do require ontology. So for example, as we enter organism, as we type through, you can see that it's linked down to an ontology, and some fields kind of have already a specified ontology. So for organism, we use NCBI taxon, and that's really just to harmonize the data as well. And like I mentioned, as you fill in your fields, some will require ontologies, and you can see that we also require you to add in units, and this also helps us harmonize as we look across multiple data sets. So if you were looking as a unit for dose, here we have gray, and we use the units ontology to make sure everybody's consistently using the right unit. So a big part of our database is, one, making sure we collect data correctly, but making sure it's harmonized. And as you navigate right through your submission process, the next step is to populate your assays. So for example, here, the first assay I added was RNA-seq assay. And you can see there's a default set of parameters that are collected with your assay, but you can always add more characteristics or parameters or comments that are associated to the assay as well, and then more samples that come from the samples table. So all this is linked as you start building. If you have an additional assay that was done within this experiment, you can click on Add Assay, and then select the type of measurement, technology, and platform use. And what that does is it builds a template that's associated with that type of assay, so you can fill in the parameters that are associated. So we've kind of configured this for quite a lot of assays and sample types in our database, and we continue to add new types as they get either developed or as they get submitted. And at the assay level, you can also link your files. So files are associated on the sample level as well. So as we mentioned, the workspace is a place where you can store your files long-term, but also once you're ready to submit your data, you can associate those files to the submission portal. And once you're got all your files and metadata in there, at the end, we do have a study validation that allows you to, that tells you that you've successfully populated the data correctly as well. So it tells you that you did a great job and that you don't have any errors. So as you're populating your metadata and data, one thing we provide is a study preview, and this is private to you. So this allows you to see what the data will look like in the in the format that you see in Genelab data repository. So you can look at the study preview at any time. We encourage PIs to do this before they send it back before they request for it to be published, so they can make sure that they populated the data correctly and it looks like how they want it. Another option here is you can share this private link with reviewers and collaborators. So in the top of your submission, there is a link that has a special access code that allows you to share this privately. So when you're submitting your paper and the reviewer is required to see that you've submitted your data somewhere, you can share this link with them, and the collaborators So this is an option that we provide for people that are submitting papers or people that have co-collaborators. So this is an option for all our submitters, and the link is private. So once you're done with your submission and you're ready to make it public, you move the step over to review, and our curators at Genelab will make sure that all the data is populated correctly, and then they will put it on the public repository on the date that you request. So that's kind of the overview of our data repository and our submission portal on how to get data into Genelab. So if you do have some space relevant omics data, please go through our submission portal and get the data into Genelab. And then if you're just interested in seeing the type of omics that's been generated from space relevant data, check us out at in our data repository. Before we finish, I do want to give you a little sneak peek on some of the tools we're working on, and this is related to visualization, and this really allows you to explore the data even more. So to get started, environmental data, this is really important to really support additional kind of metadata and supporting data for our space-flown experiments. So as Sylvain mentioned, we have radiation dosimetry data from STS, ISS, and some free flyers. We also have some ISS cabin CO2 temperature and humidity data that's associated with the omics data that's already in Genelab. So currently, if you click on the environmental data, it takes you to this summary table that provides you information about the data that's collected, and then the summarized level of data that's associated to each GLDS. So it is a really cool tool that supports the omics data that we currently have. In addition, this summer, one of our interns worked on an app to visualize the environmental data, and this is coming soon. It's not publicly available, but I just wanted to give you a sneak peek on what that looks like. So here it's very similar. You can choose the mission you're interested in, or the GLDS you're interested in. So for example, here we're looking at a rodent research 1 mission, and it gives me a summarized table of all the values for temperature, relative humidity, CO2, and radiation. And here you could see that it also gives you some time points or important dates about your launch and sacrifice dates and the duration of the experiment. In addition, we get to the plots. So it's a very interactive visualization tool. So you can look at different types of plots. So you can look at a time plot or a column chart. You can select or unselect the type of factors you would like to see. If you want to see if you want to compare it to the ground control data that is replicated here, if you want to display the different periods throughout the mission and experiment, and also some do allow you to turn off light and to associate the time periods that light was on and off. So it is very interactive. You can choose what you would like to see. So for example, here we have plotted temperature, humidity, and CO2 on the same plot. And another option is, because we like it so much, is the ability to compare across data sets. So for example, we're only looking at radiation, and right now we have an example of the GCR daily dose for RR1 mission, and you could see that across time. And if I was interested in adding another mission or changing the plots that are displayed, I could do that using the left menu as well. So I'm interested in adding RR3 to compare it to what RR1 looked like. And you could see both are plotted here with RR1 in blue and RR3 in orange. And we're looking at the time plot for the daily dose. And at the bottom, you can see we also have a plot to look at the accumulated radiation. And you can see that RR3 is a little bit longer than RR1. So it gives you a good visual of the radiation collected during that time period. Okay, so that will be coming soon. Another part of our visualization portal and capabilities is visualizing the process data that's currently in Genelab. And this tool is available. So as I showed you earlier on a data set, if you click visualization on the left menu, it will take you to a portal that has visualization on a per study data basis. So just as an example, here you could see that we have a PCA plot on the very left comparing spaceflight and ground for this GLDS 104 data set, a volcano plot in the middle, and a heat map over to the right. And you can navigate through the tab up top to look at all the other visualization plots and tables that are available. What we're working on and developing in-house is what we call Visualization 2.0. And this adds new types of visualization tools, and it also allows you to compare across multiple data sets. So this is just an example of how you would do it, and we're still building on that, and hopefully we'll have it out soon. So an example is you can use these pie charts to kind of filter down what kind of studies you want to look at. And as you filter down, it'll allow you to pick the other factors as well. So if I choose a factor for spaceflight, I can choose the organism I would like to see and tissue and so on. And once you select down to what you want to see, it will populate a list of all the available data sets that are based on your filter search. And then if you want to select all of those studies, you can do that and compare them, or if you just want to select just a few, you select a few and you click load study. So for our example today, I've selected 3 studies from the RR6 mission, and you can see that here on the left-hand side. And the first plot you're going to get here is the PCA plot. And this is very, very interactive. You can choose the different factors and the different types of the different factors that you would like to see on the graph. You can also change the color of the factor you would like to associate to the PCA plot, and you can toggle between a 2D or 3D plot as well. Another visualization tool that's associated here is the query data, query table, as we have, and that gives you the results of the differential expression, and you can change your parameters here. You can also use this to search for your favorite gene or the gene that you're interested in. So for example, here we're looking at CAV2, and we're looking at it across the 3 data sets, so you can get the log-fold change, the adjusted p-value, so you can see how this gene is changing across the different data sets. And then we also have pair plots, volcano plots, and heat maps associated as well. Part of the new tools is the ability to do pathway analyses using gene set enrichment, and this allows you to do that on a per data set level, but even across data sets. So you can choose your parameters on the left-hand side, choose the data sets you would like to load, and then run GSCA, which will give you kind of a few different outputs. So you can get a summary table on the pathways, you can get a network plot, a heat map for each analysis that you run. So this is a kind of a summary table of the results. And then going over to the next one is an interactive network plot, which allows you to change the parameters both on the left-hand side for network settings and the right-hand side for plot settings. So you can change it based on what you would like to see. And the plot itself is very interactive, so you can move the bubbles kind of around and take that with you to put in a publication or a presentation that you're preparing. And then if you wanted to run another GSA, it runs fairly quickly, and you can keep doing that as you look at multiple data sets or a single data set. So the new Visualization 2.0 and the environmental data app are still in development, and they're coming soon. But there is still the tools that are available currently in the website. So please check us out. If you have any feedback or interest, please also reach out to us. So in summary, you got a glimpse of our data repository, our submission portal, our environmental portal, and our visualization portal. In addition, please check out our other applications as the analysis platform and also the workspace if you need a place to store your omics data and then associate that back to the submission portal. And as Sylvain mentioned, standards is a big part of our workflow. in our sample processing, in our database, and in our data processing, utilizing our working groups that support us, and really also training this, new generation of scientists through the education working group, all kind of leading to this new knowledge that we have in space biology. So we're excited to support that, and really excited to give this talk to this symposium, and hopefully get some new scientists and new users of the data. And lastly, thank you for having us, and also thank you for our wonderful team that builds this and maintains the data here in Genelab. And Genelab is funded by NASA Space Biology, and our website is here as well, so genelab.nasa.gov. And if you're on social media, check us out at NASA Genelab. Thank you.
Fantastic. Thank you so much, both Sam and Sylvain. That was really great. And I would totally agree. I think that this presentation really helps more researchers across the globe really to get an idea of what the capabilities of Genelab is, and all the things that are coming are really exciting. And for people like me who may not have a very strong background in omics, it seems like even I one day can make use of this data and use those new visualization tools. That's really fantastic. I think in the interest of time, I'm just going to pose one quick question from the audience. Is it, so given that the space-flown samples are precious, but also, may need some special requirements in terms of how they're handled, would you, and either one of you are welcome to answer, would you prefer actually that PIs would analyze these samples with you directly rather than analyze it, whichever other way they normally do, and that way you already have this level of
Okay, I can take that.
yes.
Yes. Yes, in a sense, like Sylvain mentioned, we do have a lot of groups that support analyses, both on providing standards on the sample processing, but also on the data processing. So we do encourage folks to, participate in the ISOP to make sure that their sample processing is kind of in line or, in agreement with kind of what the overall space biology community is saying. And this is international, it's not NASA driven. So we do have a lot of input from, many different space agencies and just kind of scientists around the world. If you are a NASA PI or has NASA funding, it is something that Genelab can perform. They can perform the sample processing for you. But I think that, our SOPs and our pipelines are all publicly available, just like our data. We really push on open science. So if you would like to see SOPs or, data processing, how we do it, that is publicly available, and you'll find that on our website. And if you don't, please reach out to me. And the same on the data processing, we do have standard pipelines that we do use, and our AWGs also use. So yes, we do support Fantastic. That. And if it's your first time and you just really haven't worked on spaceflight data before, our analysis working group is, have a lot of space biologists that are experts and bioinformaticians that are experts that can help you walk through your analyses as well.
Fantastic. Well, thank you. Thank you both very much for your time and for joining us here. And we'll get really a big crowd that will watch this and the recording. And so, yeah, with that, I hope that you all have a good rest of your day. And thank you again.
Thank you so much.
Okay. Bye.
Bye-bye.
Bye. Thank you.